Neuroscience is the physical, the hard science aspect of psychology. So when we get into neuroscience, we're getting a lot into neuroimaging, we're getting a lot into examining brain tissue itself. When we started looking at the human nervous system, even as a very young species without a whole lot of technology, we were still able to examine the brain itself. One of the first and earliest and most obvious ways to examine human brain tissue is to examine autopsy tissue, in one form or another, the tissue of a dead brain. Now, this works fairly well. You can cut it up. You don't have to worry about hurting anyone. You can cut it up. You can look at all the bits and bobs inside and examine them and see how they look. And in fact, a lot of our names for things in the brain come from this simple examination of autopsy tissue. The substantia nigra, for instance, means the black stuff. They found a small black object in the middle of the brain and named it the black stuff. We didn't know what it did at the time, so we just named it based on what it looks like. However, one of the biggest problems with examining autopsy tissue is simply that we don't know what anything does. Nothing's working. And you can't take it apart like a clock and figure out what all the bits and bobs do, because the brain is so much more complex and so much more varied than that. We actually need a living brain to examine how things work. And we can do that by testing the behavior of patients with brain damage. We can look at exactly what portions of the brain have been damaged, what portions aren't working, what portions have been removed, and we can see by examining the behavior and thoughts of the individual what certain areas of the brain do. Now, this isn't entirely true. For instance, there are several parts of the brain that govern things like breathing and heart rate, digestion, blood pressure. Various portions of the brain deal with all of these critical life-sustaining functions. If you damage any one of them, the patient dies. And so there's no way to examine exactly what those pieces do, simply examining brain-damaged patients. You have to go a little bit deeper. With the relatively recent invention of electricity, we were able to examine the electrical currents that actually pass through a functioning brain. We do this by using an electroencephalogram. If you ever see a picture of a person and they kind of have a net over their head that has all these wires coming out of it, and they all connect to a machine, and they give you like brainwave patterns, right? They actually show like little green lines or lines on a piece of paper that show you the brain waves. That is an electroencephalogram, and it records the brain activity from the surface of the scalp. The problem with an electroencephalogram is that it's very hard to pick out an individual aspect of the brain that you're actually looking at. It's impossible to see exactly what the brain's doing. All you can really see is the rapidity of neural signals that are passing through. And you can only look at the surface tissue. You can only essentially look at the neocortex area of the brain. You can't look into the hindbrain or the midbrain almost at all with an electroencephalogram. It also doesn't give you a picture of the brain. So, with some more technological advances, we begin looking into neuroimaging, first with the CT scan, which is essentially an X-ray of the brain. But we can also use positron emission tomography, or a PET scan. We do this by creating a radioactive sugar, and we inject it into the body. Now, the brain uses almost a third of the sugar that is processed by the body. So an enormous amount of this radioactive sugar is processed by the brain. As it's broken down into energy, it emits positrons as part of its radioactive signature, and we can map those positrons and take still pictures of the brain to see what parts of the brain are doing what. MRI and fMRI machines were further advances and far more economical, because you don't have to produce a short-lived radioactive sugar in order to make them work. Instead, an MRI and an fMRI machine are both enormous magnets, and they're incredibly powerful. someone took a decommissioned MRI machine and used it to tear an office chair apart. Now, humans are not magnetic, so we don't have to worry about that unless you have pieces of metal in your body, in which case you can't do an MRI. Instead, we have to look at a different method. There are newer methods of neuroimaging, including magnetoencephalography, which is similar to an MRI using a magnet to take pictures of the brain, and the SQUID machine, the superconducting quantum interference device. That's a SQUID machine. And using the SQUID machine, we have made enormous leaps ahead in finding out how the brain works, and actually how this quantum interference helps the brain process information in a method that we couldn't have even conceived of 50 years ago, let alone mapped. Other advances in our technological ability have allowed us to magnify areas of the brain and realize that it's not a homogeneous tissue, but it is rather made up of individual cells. And we can now examine those cells and see how they work in addition to simply what they look like. The classic nerve cell is a neuron, and we'll look a little bit more in depth into neurons later on in the chapter. But there are about 100 billion neurons in the human brain. Consider that there are about 7 billion people on the earth. There are 100 billion neurons in every brain. Okay, they are arranged into networks, with each of those neurons having literally thousands of connections to the neurons around it. It is an enormous amount of information that can pass through these networks. The brain is also composed of glia cells, or glial cells. These cells are generally considered to support the neurons. For instance, the astroglia are probably the most common and actually do a good number of different tasks. There are several kinds of astroglia, for instance. One kind is actually a stem cell and helps create new neurons, rebuilding the brain when it is damaged. Another form of astroglia we believe is actually the root processing system in the brain. We used to believe that it was neurons that were the root processing system, but it's simply not complex enough to do the amount of information processing present inside the human brain. Instead, we believe that the brain is actually a quantum computer using these glial cells, and neurons are exactly what they appear to be, conductive wires that allow communication between these glial cells. Oligodendroglia help form the myelin sheath around the neurons, allowing rapid communication and advanced neural networks. And microglia clean up dead cells and prevent infection. It's important to note that things like white blood cells cannot ever interact with your brain cells. Your blood is actually toxic to your brain in a great many ways, and therefore we develop a blood-brain barrier. Okay, and these microglia are part of, and some astroglia are part of that blood-brain barrier, preventing the blood from interacting with your neurons. As a result, your brain doesn't really have an immune system. If something gets into your neural tissue, for instance, HIV or syphilis can both invade brain tissue, there's no way for your white blood cells to attack those infections. And so they can eat away your brain freely, except that these microglia, to a certain extent, do help prevent those infections until they become overwhelming. Neurons are by far the part of the brain that we know the most about. They're extremely large and easy to examine, and so we know a great deal about them. You can see in this diagram the cell body is labeled as A. It is the large bulbous part of the cell, just like any other cell. It has nucleus with DNA and Golgi bodies and all of those wonderful organelles that you learn about in biology. This provides energy for the neuron and programming for the neuron, which may actually be far more complex than we could have ever imagined. The dendrites are short antennae, sort of, that extend off of the cell body and receive messages from other neurons. Dendrites always receive messages. They can never send a message. They can only receive. But they are connected to perhaps thousands of neurons all around that cell body. In a gap in the dendrites extends a long wire-like tendril called the axon. This is labeled as C to a certain extent, also F. It's the area that's surrounded by this pink tissue. The axon carries information away from the cell in the form of an electrical signal. Labeled as D, the axon terminals actually transmit those signals to the dendrites of the surrounding cells, allowing the signal to be transmitted and to complete these neural networks. Down on this inflated view, you can see that E is actually pointing to this pink tissue that surrounds the axon. That's the myelin sheath. This is the substance that actually speeds up the firing of the neuron. This is a fatty tissue that surrounds each of the axons, allowing them to fire. It's important to note that this is a fatty tissue. Completely removing fats from your diet can prevent myelin sheath from forming or even cause your body to attempt to consume the myelin sheath. Individuals who experience extreme anorexia nervosa, for instance, we have found that a significant portion of their central nervous system becomes demyelinated. That is, the body has attempted to consume the myelin sheath to provide energy for the brain. The nodes of Ranvier, which are portions of the axon that are not myelinated, and they're not myelinated on purpose. They are gaps inside the myelin sheath, and they exist to allow the passage of sodium and I just lost the term. That's all right. Primarily, they allow the passage of sodium into the axon because sodium is the material that allows that electrical signal, potassium, that's the other, sodium and potassium. These two chemicals allow the passage of the electrical signal through the axon. Since it's not powerful enough to use an electrical signal the way a copper wire might in your house, it uses sodium and potassium instead, and it has to be regenerated from time to time. So the nodes of Ranvier allow that to happen. As I said, a neuron is not big enough and doesn't transmit enough power to transmit energy the way a copper wire in your house would. Instead, they actually have sort of a reverse, where your brain, any given neuron, while it's at rest, actually has a negative charge, unlike the wires in your house that have a negative charge when they're active. The cells of your brain actually have a negative charge when they're inactive, and it's an incredibly small charge, a fraction of a micron of an electron volt. Very, very small amount of electricity. But the action potential reverses that, flipping the potassium and sodium ions in a rapid succession down the length of the axon. This allows a positive electric charge to run down the line of the axon, producing a shift in the electrical charge and triggering the axon terminal to release neurotransmitters into subsequent cells. So the action potential runs down the axon all the way to the axon terminal. This triggers the release of neurotransmitters. There's not enough electrical signal to transmit energy from one neuron to the next, even in this incredibly fine gap between them. As a result, they have to use these neurotransmitters, these physical chemicals, to transmit energy from, or to transmit signal from one neuron to the next. These chemicals are ejected from the axon terminals, and float across what we refer to as a synapse, which is a small gap between the axon terminals and the receiving dendrites of the next cell. So they float across and touch the receptor sites of the next cell. Now, any given receptor site can only receive one form of neurotransmitter. The other forms of neurotransmitter won't stick, and so they won't create a signal in that receiving cell. So the receptor sites allow the binding of neurotransmitters and allowing a signal to be transmitted to the receiving cell. Synaptic plasticity talks about it is the process by which repeated stimulation causes a permanent change. So the more stimulation is received by the dendrites of the receiving cell, the easier it is for that cell to be triggered by reception of that dendrite. This is what we used to think actually produced the process of learning in the brain. Now we know that it actually produces something more like priming. It's a relatively short-lived period. We used to think that this synaptic plasticity lasted essentially forever, but it's actually a little bit more short-term than that. But by re-triggering it, you sort of re-up that synaptic plasticity, causing that particular neural network to respond faster and more accurately in the future. So this is how we get priming that we talked about during the learning chapter. Each neuron also has a refractory period. A refractory period is a period in which the neuron is not capable of firing again. Now, in neurons, a refractory period is actually extremely small, fraction of a second. Okay? But after firing, there's a short period where the neuron cannot fire again. It's not possible. There's not enough energy in the cell, and there's not enough neurotransmitters in the cell to allow it to occur again. Instead, the cell has to reuptake a certain amount of the neurotransmitters and sort of recharge in order to fire again. During this recharging phase, the cell can have a relative refractory period where the cell can fire again, but it requires a much greater amount of energy for it to work. Neurons do not function by themselves, but rather have to accumulate into neural networks. This is, I believe this is an electron microscope rendering of a very simple neural network, where you can see the neurons grouped together actually very, very closely and so interconnected like this. These clusters of neurons all communicate with one another, and any given network is connected to other networks. Any given cell can be connected to a multitude of networks. These networks are extremely complex and work in ways that we still haven't completely mapped out. Just because with 100 billion neurons, it's almost impossible for us to deal with the sheer number of networks that exist within a normal, healthy adult brain. But these neural networks exist and provide the complexity of networking that's necessary for the human brain to process information. We can look at how the nervous system is organized beyond just the cellular level, beyond just the neural network level, and actually look at the broader aspects of these tissues. The central nervous system is fairly simple for these purposes. The brain and the spinal cord. That's the central nervous system, and that's all the central nervous system. Everything else, including, for instance, your optical nerves, they're still in your head, but not a portion of your brain. But only the brain and the spinal cord are your central nervous system. Everything else is your peripheral nervous system, right? And the peripheral nervous system represent all of the neurons that travel throughout the rest of your body. They represent the neurons that bring signal in, and they represent the neurons that send signal out. The somatic nervous system okay, is essentially your sensory nervous system. It is all the neurons that gather the sensory information and bring it from all over your body to the spinal cord and brain. Your optic nerve, the nerves in your skin that let you feel things, your taste buds, your olfactory buds, your hearing hair cells, all of these are aspects of your somatic nervous system. And the somatic nervous system only goes into the brain. There's no aspect of the somatic nervous system that goes out. Okay? And the somatic nervous system does not overlap with the autonomic nervous system. It requires a whole different set of nerves. The autonomic nervous system sends signals out from your brain. Okay? So the autonomic nervous system always sends signal out. And there's two non-overlapping sets of nerves that send signal out from your brain. Remember, these do not overlap. You have the sympathetic nervous system and the parasympathetic nervous system. Both of these are part of the autonomic nervous system. The sympathetic nervous system, most of the time, the sympathetic nervous system turns things on. The sympathetic nervous system makes your heart beat faster. The sympathetic nervous system makes your muscles contract. The sympathetic nervous system makes your lungs breathe. It turns all these things on. However, the sympathetic nervous system also suppresses digestion. It suppresses your sex drive. It suppresses several of the other things that you really only do when you're relaxed. The purpose of the sympathetic nervous system, the general purpose of the sympathetic nervous system, is to prepare you for a fight or flight situation, for a panic situation. The parasympathetic nervous system does exactly the opposite. It slows everything down. It slows your heart down. It slows down your breathing. It allows your muscles to relax. It also activates your digestive system. It activates your sex drive. So the parasympathetic nervous system and the parasympathetic nervous system and the sympathetic nervous system work in parallel and opposite to one another. So they counter one another. If only one of those systems were working, you could not survive, because it's necessary for your heart both to contract and to relax. And your sympathetic and parasympathetic nervous system work to balance that and keep your heart beating at a steady rate, to keep you breathing at a steady rate. All right. They also allow all of your muscles to contract and relax so that you can have voluntary and involuntary motion that's necessary for your existence. This is a fun little diagram of your system with the central nervous system there in yellow, and then you have generally, I believe, the somatic nervous system in blue and the autonomic nervous system in pink. It's of course a grossly incomplete drawing, but it's sort of an example of what you might look at in those three systems. So the structures of the brain, because the brain is by far the most complex organ that you have. It's composed of the brain stem, which has, we're just going to skim through all of this because we're going to come back to most of these here in just a moment. We can look at all these different structures. Here's a fun little picture. You can see the brain stem here, and the cerebellum, which is the little brain right here. This is the midbrain area, which has all the little tiny pieces. And then this wrinkly section on top, that is your cerebral cortex or your neocortex, depending on the terminology you choose to use. The brain stem is by far the simplest aspect of your brain, sometimes called your reptilian brain. This area of the brain is actually even simpler than most reptiles, even though, of course, reptiles also have it. The brainstem regulates basic life functions, things like heart rate and breathing, digestion. It prevents you from hiccuping, prevents you from vomiting, that sort of thing. The brainstem is essentially a swelling at the top of your spinal cord. It's a more complex section of your spinal cord. It includes things like, for instance, the reticular formation, which regulates your sleep-wake cycle and is the source of melatonin in your system. It's also a source of serotonin, which is important for mood and activity levels. These are neurotransmitters. The pons is a larger swelling at the vast majority of that swelling of the brainstem. Produces norepinephrine, which keeps you alert and awake. That gives you sort of panic mode. The pons allows you to have that fight or flight response. The medulla regulates your heartbeat, breathing, swallowing, coughing, that sort of thing. It keeps you from choking on your own tongue. Which is kind of horrible, but it's necessary. And these are the most basic aspects of the brain. These are what is bare minimum necessary for an organism that has a central nervous system to operate. The cerebellum is a small lump of tissue, a little bit smaller than your fist, right at the back of your skull. And it actually looks like a tiny brain, and that's what cerebellum means, is tiny brain. We actually think that this helps control basic motor coordination, particularly gross motor coordination, like walking, for instance. The cerebellum is a relatively simple form of brain tissue, and we think it was sort of the original neocortex that continues to remain functional, and so it's kept evolutionarily. As we move forward and into the very depths of the brain, we reach the area called the midbrain, which actually does a lot, right? This is the part with all the little tiny bits and bobs in it that we can actually take apart and look at. The thalamus, for instance, functions as the brain's telephone operator, sort of. It accumulates message from the sensory nerves and distributes it throughout the brain to the relevant sensory areas. The hypothalamus, which means the piece below the thalamus, and it is below the thalamus, it helps us with eating, drinking, body temperature, blood chemistry. It maintains your body's equilibrium for the most part. The pituitary gland helps to regulate hormones throughout your body because neurotransmitters can only exist inside neural tissue. In order to transmit information to the rest of your body, we use hormones, which are chemically almost exactly the same, but exist in your bloodstream rather than between neurons. The limbic system is what produces most of these hormones as well. The amygdala provides control over fear and aggression responses. Your hippocampus is heavily involved in regulating memory. The striatum allows you to use fluid motions and helps you with unconscious learning and memory, things like procedural memory. And the nucleus accumbens, for instance, deals with motivation, reward, and addiction. This is the source of most of your dopamine cycle. This is just an example of some of the things that are present in your midbrain. There are dozens of little bits that exist in your midbrain, and you don't really need to memorize them all unless you plan to be a neurosurgeon or a psychiatrist, in which case you should probably know rather more about the midbrain than, well, frankly, than I do. So this is sort of an overview of the midbrain, which is relatively small but very complex. The neocortex is the wrinkly gray and white tissue on top. It's the outer surface of your brain and provides the majority of the surface area of your brain. It also deals with the most complex parts of your behavior and thought processes. The frontal lobe, including the prefrontal cortex, deals with most of your higher intellectual processes. This is behind your forehead and behind your eyes, and it's the most forward section of your brain. It's the reason humans have this large protruding forehead, whereas most of our simian relatives have a swept back forehead. We need a larger frontal lobe area. The frontal lobe controls things like planning ahead, hypothetical reasoning, compassion, and love. It also helps us produce speech, working memory, morality, and mood. This Broca's area is just in front of the temporal lobe on the left-hand side and helps with speech production. The occipital lobe sits at the very back of your brain, and it deals exclusively with vision. The occipital lobe does nothing else. The parietal lobe exists from the crown back to the occipital lobe and deals with your perception of touch and to a certain extent with your motor functions as well. The temporal lobe deals with hearing, language learning, memory, spatial reasoning, that sort of thing. Weinicke's area exists directly behind Broca's area and deals with language comprehension. This section of the brain deals with all of those wonderful speech and hearing and understanding aspects of your brain and your neural networks. There you can see a nice little drawing of the four major lobes of the brain, with you can see the cerebellum sticking out of the bottom as well. Inside the cerebral cortex, there are a bunch of sections of the brain that do all kinds of wonderful things. One of the largest sections of the cerebral cortex is this primary sensory strip and the primary motor strip. These two strips run right across the crown of your head from the tip of one ear across the crown to the tip of the other. And they are responsible for most of your touch sensory information, as well as most of your voluntary motion. And it's sort of balanced between the frontal and parietal lobes. But these sections of your brain control an enormous amount of your exterior behavior. The association cortices, and there are multiple association cortices, allow complex functions. They allow you to integrate information from a variety of sensory functions. Without an association cortex, you couldn't fully enjoy a meal, for instance. Enjoying a meal often deals with taste and smell and touch for the texture in your mouth, and sight to see the presentation of it. A meal is an extremely complex sensory experience, and these association areas allow us to combine all of those sensory pieces into a single experience that we can think about and talk about and have. This is your corpus callosum. You see in the middle this little Y of tissue that's very white, very highly myelinated. This extremely dense area of neurons that connect the two hemispheres of the brain, which you can see are actually physically divided. This particular brain is split essentially from ear to ear, so you're looking from front to back in this image. But the corpus callosum allows communication between the two hemispheres of the brain. Without this, the two halves of the brain don't communicate very well, but we have actually, in certain people, cut the corpus callosum and severed those lines of communication. Not just to do it, but rather because the corpus callosum is also the part of the brain that allows massive seizures to occur. Without the corpus callosum, the seizures stop immediately, but it also produces a set of interesting side effects, including the ghost hand effect. But we don't have to do these split brain procedures anymore, since we now have a much better anti-seizure medication that you can just take a pill and prevent seizures in the future. So everything we know about cutting the corpus callosum is, for the most part, said and done. Those aspects of research are complete, and we don't have a reason to do those studies anymore. This is an example of how neural tissue is actually developed. This is an example of an embryo. It's already been implanted in the uterine lining. But a certain portion of the tissue of that embryo begins to fold in on itself, creating a tube. And this is the very beginning of the spinal cord that will eventually run down the center of the back. The top of this neural tube begins to split and divide and becomes the two halves of the brain. And they grow. It actually grows extremely rapidly, starting at about the third week of prenatal development. This neural tube is the beginning of the brain, and its split represents the two hemispheres of the brain. During this period, we undergo a process called neurogenesis. That's actually the creation of new neurons from stem cells. These neurons are created inside the neural tube and migrate up into the new brain tissue that's being developed. We found that we actually can't interrupt this process. It's not possible. Even removing a single neuron that's undergoing migration and moving it even to the other side of the neural tube, this single neuron will then migrate back to its original position and then continue on its way. We can't interrupt this process, and we don't know why. We're still not entirely sure how this neural migration works, except that it does, and that it produces a unique brain structure for every individual. Now, that's not to say that every individual has different structures in their midbrain and that their neocortex is organized and designed differently, because they're not. If you cut a brain up and examined it, they would all look essentially the same. But under an electron microscope, you can see that everyone's neural networks are unique, completely unique, to the order of hundreds of trillions, one in hundreds of trillions. Where you're never going to find two brains that are exactly the same. We also undergo a process called synaptogenesis, which is creating new networks and new connections between those budding neurons. Because your brain is divided up into two halves, it's very reasonable to begin to assume that those two halves perform different tasks. And to a certain extent, they do. We refer to this as brain lateralization. This tends to differ between individuals. Some people are more lateralized than others. That is, the parts of their brains are more specialized, or that the halves of their brain are more specialized than other people. However, we do know that women tend to be less lateralized than men, and left-handed men tend to be less lateralized than right-handed men. In general, though, the left half of the brain can accomplish anything that the right half of the brain can, and vice versa. The right half of the brain can do anything the left half can do. The issue is that specialization and thus lateralization makes certain areas of the brain more efficient. For instance, why would the right half of the brain focus on learning language when the left half of the brain already has it covered? The right half of the brain can dedicate that portion of its area, that portion of its volume, to doing other tasks, visualization or spatial reasoning, for instance, instead of language acquisition. When a portion of the brain is damaged, however, the other half of the brain can sort of pick up the slack and take up whatever is needing doing. It's just a little bit less efficient at doing it. There have also been instances of people born with only one half of their brain. There was, for instance, a few years ago, a paper done on a young woman who had been born with only the left half of her brain. The right half of the brain was filled up with cerebrospinal fluid instead of brain tissue. She was perfectly proficient, graduated high school. She wasn't necessarily valedictorian, but she was just fine, and no one ever noticed that half of her brain had never formed until she had a traumatic head injury and had to go in for a CT scan. Simply no one noticed. She was just fine, an average student with no other problems. She just had half a brain. So brain lateralization works on a point of efficiency rather than necessity. There is very little relationship between brain size and IQ. On the whole, most human brains are approximately the same size, no matter what size your head is. Some people's brains may be a little bit bigger, a little bit smaller, plus or minus 5% from average usually. If you get out beyond about 10%, either too large or too small, you start having mental deficiencies, you start having problems. A brain that's too large has problems almost identical as brains that are too small. And having a particularly large brain has no significant advantage. There have certainly been reports that Einstein's brain was larger than the average brain, um, but it doesn't help, right? Having a particularly large brain doesn't give you any unique advantages over other people. Neurological diseases are, of course, biological diseases of the brain. We can have what we refer to as a psychiatric illness, which is different than a psychological illness. A psychiatric illness presents as a psychological illness, like, for instance, depression, except that it's caused by a biochemical imbalance. It's actually caused by a physical problem inside the brain, usually too many or too few neurotransmitters. And as a result, we can treat these with medication. Neurological illnesses are caused by structural problems in the brain or occasionally by nerve death. Multiple sclerosis, for instance, is caused by the lack of myelination or the loss of myelination, which causes poor motor skills, poor sensory capabilities, poor cognition, that sort of thing, and a lot of pain. ALS, also called Lou Gehrig's disease, is the gradual death of motor neurons, which prevents the individual from moving with any kind of accuracy, skill, or strength. Parkinson's disease is the destruction of the dopamine neurons. I'm not going to try and read that word as it is. The dopamine neurons begin to die. Okay? This causes tremors, it causes dementia, it causes difficulty feeling pleasure. Huntington's disease causes death in the neurons of the striatum, which causes awkward motions. It causes jerky motions, symptoms of psychosis. There are a bunch of these neurological illnesses depending on what portions of the brain have begun to die. But though there appears to be a genetic link in a lot of these, we're constantly working on drugs and therapies that can slow down or even prevent these neurological illnesses. So that is our examination of neuroscience and psychiatry for the time being. So that is, and that's the human brain, and that is essentially all brains. They're all put together very similarly, except that the human brain is more advanced, particularly in the frontal lobe. So, hope you all have a good week. Thank you very much.